Organisms come together in ecological communities, comprising populations of at least 2 distinct species that interact both directly and indirectly, shaping the very fabric of life. Some of these interactions are positive, benefiting one or both species involved. Interactions can also be negative, where one species thrives at the expense of the other. Nowhere are these interactions more apparent than on the rocky shore. Negative biotic interactions are evident through competition for resources, the looming threat of predation, and parasitism. When considering negative interactions, look no further than rocky shore regular Carcinus manus, or better known as the green shore crab. Carcinus manus are voracious, opportunistic predators that feed on a wide range of organisms. Whilst native to our shores, they are invasive to others, causing pronounced declines in local populations. Here, we will focus on parasitism in Carcinus manus and its potential effects for biodiversity as a whole.
Parasitism is a symbiotic relationship in which one organism benefits while the other is harmed. The parasite benefits by gaining nutrients and/or energy from its host. Sacculina carcini is a parasite of the green shore crab Carcinus manus. This is a life cycle that shows how the parasite infects its host. Firstly, the female parasite invades a host crab that has recently molted as their carapace is softer. Once on the shell, the kentrogen larvae inject their cells into the crab's hemocell via a needle-like cuticle called the stylet. Once inside, the kentrogen larvae follow the crab's circulatory system until they reach the abdominal cavity. There, the larvae attach themselves to the host tissues and form a root-like network known as the interna. The roots grow by absorbing nutrients from the crab's hemolymph. After 1 to 3 years, the interna ruptures the crab's carapace and becomes exposed to the water. The interna is now an externa. The externa is a brooding sac which contains developing parasite eggs, which are fertilised by a male cyprid parasite. After fertilisation, the nauplius larvae are spawned from the externa into the water in search of a new host. Sacculina carcini is a parasitic castrator that disrupts host crab reproduction. Their roots grow through the host gonadal tissue, which halts sperm and egg production. This process leads to feminizing morphological changes as the crab's abdomen widens and they carry the externae underneath the pleon where egg-bearing females keep their eggs. This is to attract a new male host. There are also potential behavioural changes, such as migration towards deeper water, copying egg-bearing females. In 2016, Weiser et al. Investigated this by sampling crabs at 12 locations in the Dutch Wadden Sea at 3 different depths. At each location, they compared the distribution of infected shore crabs to the distribution of ovigerous or egg-bearing females. They found that both the prevalence of infected crabs and of ovigerous females significantly increased with water depth, with the p-value being less than 0.01. The results suggest parasite-mediated behavioural changes, where the infected crabs behave like ovigerous females, migrating towards deeper waters.
Sacculina carcini significantly reduces the population of Carcinus manus by impairing their reproduction. Despite having negative effects for the crab itself, there are benefits for prey population dynamics. Carcinus manus is a highly invasive species which consumes native species such as eelgrass, which is important in habitat formation. If the population and distribution of Carcinus manus remains stable, it allows native biodiversity to thrive.